All right, translation, the second half of protein synthesis. Block day, we talked about the transcription process. So if you think about what transcription means, it is essentially repeating or restating, making a copy of something else. So I'm watching a movie and I want to put closed captions or subtitles on. It's just telling me what's being said. It's just copying the things that those people are saying, making it into a written language. Same language, just a different copy. So that's how I kind of tell the difference between transcription and translation. DNA and RNA are both nucleic acids. There isn't a lot of translation that needs to happen there. We're just making a copy. However, in this second part, we are actually doing a lot of translation. Two completely different languages. We have nucleic acids and proteins. Different macromolecules, completely different language. At least between DNA and RNA, they're still the same type of macromolecule. Not a lot of difference there. It's maybe like the difference between American English versus British English. Slight differences, but we can still understand each other. We're good to go. Translation from this nucleic acid language into the protein language, a lot more barriers. Different macromolecules. That's like American English versus like Taiwanese. Completely different. So translation is this more complicated process of making that understandable to what our protein needs to become from that transcription of mRNA. All right, pointer out. All right, so here's a brief, short breakdown. At the top here, we have DNA. That's our original strand, our OG code, ready to go for whatever protein this is building. Then what we learned about on block day, our mRNA makes a transcript, it transcribes that information, makes a copy. But the question is, how does that RNA language turn into a protein? Where did those amino acids come into play? There's only 20 of them. How does it make a protein? So looking at our mRNA here. mRNA codes for proteins and things called codons, triplets. So for every 3 base section, little nucleotides and sets of 3, those are called codons. So as things are read, each set of 3 or triplets or however you want to call it, of bases, nucleotides, letters, that's a codon. Proteins are made of amino acids, and those amino acids are recognized by that codon. Each codon is a sort of word to signal, hey, we're making this amino acid. Here's another way to say it. Repetition is key here. A codon is 3 base pairs. So 3 of those nucleotides, that means, hey, we're making this amino acid. So the amino acid needs 3 pieces of information, 3 of those base pairs, to be recognized, to say, hey, we're making this. That grouping is called a codon, 3 of them altogether, nitrogen bases, nucleic acids, 3 of them make a codon. All right, so this is called the code for life. What does that mean? Well, to make life, we need proteins to do the work to make the organism function. And to make proteins, we need amino acids. Amino acids are the key here. Without amino acids, life can't exist. Think about that for a second. Without amino acids, we cannot have life. So we can have all the DNA we want, but if we can't turn that into a protein, it's not going to turn into a living organism. Something else we should note is that the code is redundant. There's only 20 amino acids and way more combinations of these nucleotides. So there's going to be some repeats. The same amino acid will have a different number of codes. What do I mean by that? For example, let's take this leucine, for example. So first column, second row, all 4 of these combinations of nucleotides, all 4 of these different codons have this, they make the same amino acid, leucine. So if our ribosome is reading this mRNA, CUU, CUC, CUA, CUG. Anytime it sees that, hey, we're making a leucine. Doesn't matter which one, I know I'm making leucine, so there's going to be some repetition. There are 2 amino acids that only have one sequence. One of them is tryptophan up here. Not that important that you know that there's only one codon for that. But the most important one, in my opinion, is this start codon, AUG, or methionine. I can never say it right. It's always shortened to MET. All of these amino acids are shortened to 3 letters to help, just make the process easier and quicker. Don't have to worry about all of those weird sounding complicated words. So the start codon, AUG, or methionine, if I'm saying that right. This is a signal. Hey, you see me, you see this code, see this amino acid? We're gonna start here. And everything after me is part of the protein. Everything before me doesn't matter. I am the start. I'm going to be the beginning of this protein. Then after that AUG start codon, we just chug along through these codons, attaching amino acids until we get through our mRNA chain, and we hit something else called a stop codon. Stop codon means, hey, we're stopping. We don't need any more. Our protein is as long as it needs to be. We have all the right amino acids in order. Our protein is done. It's finished. So we're going to stop the coding process. We're going to stop attaching amino acids. And there goes our protein, ready to move on in the process, usually through that endoplasmic reticulum to fold it correctly, then to the Golgi apparatus to package it, ship it where it needs to go. So a codon chart. We talked about codons, what they do, how they work, but I don't expect anyone to remember or memorize what codes are for what amino acids. That's ridiculous. So we have these tools, one of which is called a codon chart. Pretty easy. You take your first letter from this side over here, you take your second letter from the top, and your third letter from the left. So what does that look like? For example, we have a codon GCA. How do we know that that is a code for the amino acid alanine? Well, take the first letter G, find that on the left. Then the next letter C, find that on the top. All right, so we know we're in this little quadrant here where those two intercept. Find the last base in there, A, on the right, where those 3 intersect. That is going to be our amino acid for that codon. Another example, UUC. All right, starting on the left, find that U. Great, we're going to be in this row. Another U. Awesome. Up in this top quadrant, we don't know if it's going to be phenylalanine or leucine. That third one far on the right is a C. Going across where they intersect gives us that amino acid. You will need to know how to use this codon chart. Another tool we have is a codon wheel. This is my personal preference. I think this is much easier to use and understand just for me personally. All you have to do is you start in the middle, work your way out. So for example, for the codon CAG, how do we know that gives us glutamine? Start in the middle, find the C, awesome. Now we're going to be in that C quadrant. What's the next one? A, great, find the A. Now we're going to be focused in that A section. Find the last one, G. That lets us know that the amino acid is glutamine. One more example, GGU. Start in the middle, find the G. Awesome, gonna be in that quadrant. Next one is another G. Awesome, great. Now we're gonna be in the G section. And then the last one, U, lets us know that it is glycine. You will need to know how to use both of these, both the chart and the wheel. Feel free during assignments, whatever, to use which one you prefer. Again, I prefer the wheel. I think it's easier. Some of you may prefer the chart. But you will need to know how to use both of these for the test. All right, so now we know all about codons and we know how they tell us which amino acids are which. Now we go to the actual building process. So up at the top, we have our DNA strand, our little code sequence here for whatever protein this is building. Underneath that, we have our mRNA. It transcribed a copy of that code. Each set of 3 will be a codon. So looking right here at the top, hey, AUG, I know that's a start codon, so that's going to be the beginning of our protein sequence. Now comes in a new type of RNA. tRNA stands for transfer RNA. You could use a sort of thinking in that the tRNA happens in translation, but the T actually stands for transfer, not translate, but it essentially is doing the translation process. It's transferring amino acids to build this protein. All right, so this tRNA now reads the mRNA codon, makes what's called an anticodon, so it's the reverse of the codon, anti, matches those base pairs to make the anticodon, and says, hey, I match these, which means I'm making this amino acid. In this case, methionine, I can't say it. And I know because the mRNA codon, AUG, stands for that start codon. Great, we did that first codon, we're gonna move on to the second one. And then the third one. And as we're producing these amino acids, we're gonna bond them together to create this chain of polypeptide or our protein. Here's another look at it. DNA at the top, I separated them more distinctly into their codons. DNA make a transcription of it with our mRNA. The tRNA matches its anticodon, figures out, hey, that was a code for the start amino acid. So we're gonna put the methionine on there to start our protein and then make our way through. tRNA. tRNA actually has those amino acids. That anticodon, so the reverse of the codon on the mRNA, that matches up. So we're matching those base pairs, but each tRNA has its own anticodon, whatever is the opposite of the mRNA, so it can match up. Then it says, hey, since I'm here and I match this codon, I'm gonna pop out the amino acid. So if I match the start codon AUG, I'm gonna pop out that methionine. Then another one comes along, another tRNA. After that process is done, it can make its amino acid, bond it, start the chain. All right, so here we are, tRNA. It matches the anticodon, produces that amino acid to bloop, 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 make that chain of proteins. From our organelle unit, we know that ribosomes are the protein makers. They're in charge, they run this show. They're directing, they're facilitating, they're making sure everything is running smoothly. They say, hey, make sure you're going to the right codons, tRNA. They're made of proteins themselves. There's a small one on the bottom and a big one on the top, and they sort of come around and sandwich the mRNA and the tRNAs and this amino acid coupling process where they bond to each other to make the protein. So they're just there to create a space, to facilitate, make sure everything is running smoothly. All right, here's a really good diagram that I like a lot. I know those words are not always helpful. All right, so on the bottom of both of these diagrams, we have a long strand of our mRNA. And here come our bent tube cylinder guys, our tRNA. They have anticodons on the bottom. They're gonna say, hey, we match up here, bloop. I'm gonna produce my amino acid. Then the next one comes along, matches up, bloop, produces their amino acid, they bond. Then once that amino acid is bonded, the first one can leave. Its job is done. And it just shifts down as they go across that mRNA strand. The 3 steps: initiation, kind of self-explanatory. Things start to happen. The ribosome, the tRNA, the mRNA, they all sort of come together to start the process. Elongation is called that because we're just elongating things. We're making a long chain of amino acids to make our protein. And termination, again, is pretty self-explanatory. We hit that stop codon to say, hey, we done it. We reached the end here. We don't need any more amino acids. Our protein is finished. So just stop, release, send it off through the endoplasmic reticulum to finish the folding, purifying process. All right, here's some GIFs that I found. This is my amoeba sisters. I love amoeba sisters. I'll wait till it starts over. All right, so we have our DNA, it separates, make room for RNA polymerase to make a copy, it's transcribing. Then that mRNA gets sent out of the nucleus, the ribosomes find it, we start matching with our tRNA anticodons, they spit out the amino acids which bind and create our long polypeptide or our protein. Here's another one. It's not the complete process, but it's just to get a feel. On the bottom is our mRNA again. Then we have our tRNA. It's coming, matching that anticodon, bloop, and now it has its start amino acid from that start codon. And after this, more tRNAs will come and match up with the codons, pop out their amino acids, bond them to make our chain of polypeptide. Here's the last GIF I have for you. Ooh, it's kind of glitchy. On the bottom, we have our mRNA again. Our ribosome is that weird blobby thing. And our tRNAs come. Their anticodons match with the codons. That tells them which amino acid to make. Amino acids pop out the top, bond, creating our long chain of amino acids or our protein. And lastly, I have a couple memes for you I thought were interesting and kind of funny. If DNA says T, 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 mRNA says ah, just to match those base pairs. And then we have Khaleesi here from Game of Thrones. I think that's who she is. I didn't watch Game of Thrones. And on the left here, she has a single curlicue spiral curl thing on her hair. And on the right side, she has sort of a double spiral, double strand curl that looks like DNA.